So now we've made our way to the electron transport chain. And again, just to summarize, we talked about how we had glycolysis. The product of that was pyruvate, and what made its way into our pyruvate catabolism. Then that made its way into the citric acid cycle. And finally, the product of that, the NADH and the FADH, the NADH and FADH is going to make its way into the electron transport chain to produce even more ATP. Okay, so this happens within the mitochondria as well. Now we're still in the mitochondria here. We have these coenzymes that have been produced on every step along the way, right? So each of one of those steps that I outlined on the previous slide, they've each generated some amount of NADH and FADH. The electron transport chain is all about breaking those, converting those coenzymes into ATP, all right? So we're going to see that these reduced coenzymes, the hydrogen ions and electrons from those molecules, are passed through this electron transport chain through a series of enzymes. It's going to consume oxygen in the process, and we're going to generate ATP. Okay, so this is actually a summary of what we did, what I just did at the end of the last slide of the previous lecture, when we went through and summed up how much ATP, FADH, and NADH we had at this point from our three processes: glycolysis, pyruvate catabolism, and the citric acid cycle. So that's what this is saying here. At the end of all of these, per one molecule of glucose, we have 4 ATP that's been generated. We have 10 NADH and 2 FADH, right? And so again, this whole idea of the electron transport chain is going to be to convert this into more ATP. That's what we care about, how much ATP we get. Okay, so in the electron transport system, there are 5 protein complexes numbered 1 through 5. These are these enzymes that are doing this work. There are also 2 electron carriers, coenzyme Q and cytochrome C, okay? Again, I kind of glossed over the role of those coenzymes, the NADH and the FADH. They were being reduced throughout the citric acid cycle, glycolysis, pyruvate catabolism. And what we're going to see is they're going to be now oxidized and transport electrons to these 2 carriers. That energy in those electrons is going to be used to pump protons from the inner membrane of the mitochondria to the inner membrane space, okay, across the inner membrane. All right, so actually let's real quick let's back up and look at a mitochondria again. Okay, we have these 2 portions of the mitochondria. There's an inner membrane and an outer membrane. Right, so everything that we're talking about now is taking place in the inside this space right here. Okay, what we're now going to be talking about with the electron transport chain is pumping ions across this inner membrane into the intermembrane space. So the intermembrane space, this region here that I'm going to color in yellow, this kind of outline, this region here, is really acidic, because we have these different enzymes that are pumping protons across that inner membrane. And what that's going to do is create a gradient where there are more protons outside that membrane than there are inside, and they want to flow back into back to the middle of that mitochondria. Okay. Okay. So what we're going to see is we have these various enzymes, and we'll talk in detail here in a second, but just broadly speaking, what's going on is there are a bunch of steps which are used to pump protons or hydrogen ions from the mitochondrial matrix into this intermembrane space. So that's what these different reactions are doing. We're using the energy harnessed in the NADH and the FADH to pump protons across this membrane. and that creates a acidic environment in this intermembrane space. Lots of hydrogen ions means it's really acidic. And this is at a low pH. The mitochondrial matrix is at a higher pH, right? So there's this, what we call a concentration gradient. There's a ton of protons on one side, not a lot on the other side. So that creates this energy gradient where those protons really want to be able to flow back across that membrane. So all of these electron transport chain enzymes, they're pumping these protons out. And then we have this last step, ATP synthase. It's actually a super cool protein structure. I studied it a little bit at some point, but it's kind of like this big wheel. Anyways, what it's going to do is allow those protons to flow back across, to go back in the other direction, but it's going to harness that energy to create ATP. Right? So you have all these different electron transport chain enzymes that are creating this concentration gradient. And then you have an ATP synthase, which is harnessing that concentration gradient to generate ATP. Okay, that's the basics of the electron transport chain. That's how NADH and FADH is used to create ATP. Okay. So step one is NADH transfers electrons through complex 1 to coenzyme Q. And in the process, complex 1 starts pumping protons across that inner membrane, right? So step one, we have NADH transferring electrons to coenzyme Q through complex 1, and that pumps some protons across that intermembrane space. Okay, so that first step, we have creation of our concentration gradient. The second step, we have FADH transferring electrons to coenzyme Q through complex 2. So complex 2 again ends up transferring electrons to coenzyme Q. This step doesn't come with any pumping of protons across that intermembrane space. Because of that, we're going to see that per molecule of NADH, we actually get more ATP than we do per molecule of FADH. All right, so now step 3 is coenzyme Q transfers those electrons that it got to cytochrome C through complex 3, right? So coenzyme Q transfers to cytochrome 3 through complex 3, and in that process, complex 3 pumps protons across that intermembrane space. Okay, so electrons are being transported, starting out on either NADH or FADH. They're going through these series of complexes, and as those electrons are getting passed, they're pumping protons across this gradient, right? So they're using that energy of passing that electron in order to create this concentration gradient. Next step is we have another transfer step where cytochrome C transfers to complex 4, and that also pumps protons across that membrane. So cytochrome C transfers to complex 4. We get even more protons pumped across the membrane. And then that last step is that complex 5, which we call ATP synthase, allows those protons to flow back, utilizes the energy of that concentration gradient, allows these protons to flow back, and in the process, it harnesses that energy to synthesize ATP, right? So ATP synthase uses the concentration gradient to synthesize ATP. Okay, so if we kind of zoom in on our little chart here, or actually let's go to the bigger one here. Step 1, NADH transfers electrons to coenzyme Q through complex 1. NADH transfers the electrons to coenzyme Q through complex 1, pumping protons across the gradient. FADH goes through complex 2 to give its electrons to coenzyme Q. That doesn't actually pump any protons across the membrane. Now coenzyme Q takes those electrons, transfers through complex 3 to cytochrome C. Complex 3 pumps protons across that gradient. Cytochrome C transfers through complex 4. Complex 4 pumps protons across the membrane. You can see that this step is also accompanied by a consumption of oxygen, right? So we said that this has to occur under aerobic conditions. Here's where oxygen's playing a role. And so that's through complex 4. And then the last step is we have complex 5, which we call ATP synthase, that's going to allow those protons, going to harness that concentration gradient, allow those protons back through, and in the process, synthesize ATP. Cool. All right, so the net result of this is per molecule of NADH, because of that concentration gradient that's created, you get 2.5 ATP per NADH, right? Because you've created this concentration gradient by allowing those protons to flow back, you get ATP. It turns out that per NADH, you get about 2.5 ATPs. FADH, we said that it produces less energy because complex 2 does not do any proton pumping. So it only creates 1.5 molecules of ATP per molecule of FADH, okay? I will point out, and I've made sure that our homework is all consistent with these numbers. I will say some textbooks that you see round this up to 3 ATP and 2 ATP. That's not what we're going to use for ours, for ARC's course. We're going to stick with this 2.5 and 1.5, okay? So we talked about how NADH generation led to an indirect payout of ATP. And now we've seen that through the electron transport chain, NADH will create 2.5 ATP per NADH. FADH will create 1.5 ATP per FADH that's consumed, okay? All right, so which one is accepting the 2 electrons from NADH, right? That's going to go through, this is actually through, we can even be more specific, through complex 1. We'll go to coenzyme Q. Which one accepts electrons? Same thing for FADH, except for this goes through complex 2. Gives it to coenzyme Q, and then coenzyme Q is what passes off its electrons to cytochrome C. Okay, so knowing those order of events is going to be really important. Okay, so which one? These are all products of either the citric acid cycle or the electron transport chain. Let's see if we can specify which is which. CO2 is a byproduct of the citric acid cycle. Actually, in the electron transport chain, although oxygen is consumed, it's not converted to CO2, it's actually converted to water. FADH is consumed in the electron transport chain. It is a product of the citric acid cycle. NAD plus is what happens when NADH is used in the electron transport chain. So that's the product that we get from NADH. NADH was the product of the citric acid cycle. And ATP is actually the product of both of these here, right? We got one payout, like a small payout of ATP from the citric acid cycle, and then the electron transport chain is when we get a lot of ATP. All right, so now we're going to sum up what our ATP payout will be for glycolysis, pyruvate catabolism, the citric acid cycle, and then finally our electron transport chain. What do we get total from all of these combined for 1 molecule of glucose? Okay, so remember in glycolysis, what was formed? We had this direct payoff of how many ATP from glycolysis. The direct payout was 2 ATPs. And then glycolysis also had this output of 2 NADHs. Okay, I'll say before I move on real quick, being able to complete this chart from memory is the most important part of this whole metabolism discussion. How much ATP do you get from each step? How much comes from direct phosphorylation? How much comes from NADH, et cetera, et cetera. Okay. Okay, so then those two NADHs enter into those two NADHs enter into the electron transport chain and yield how many ATPs? Remember, it's 2.5 ATP per NADHs, so that would be a total of 5. So from glycolysis, from both the direct phosphorylation, our direct payout of ATP, and the indirect payout that comes from those coenzymes, we would get a total of 7 ATP. Okay, pyruvate catabolism didn't generate any ATP directly, but we did get 1 NADH per pyruvate. And since glucose is converted into 2 pyruvates, we would get 2 NADHs. So then what would be the ATP yield once we get to the electron transport chain? That would create 5 ATPs. Okay. In the citric acid cycle, I told you don't have to know all the individual steps, but we should be able to figure it out for the summary here. Remember, we had 3 NADH produced at 3 steps. Oops. Right? And since we have 2 turns of the citric acid cycle, right? We have 2 turns because just like we had 2 pyruvates created from 1 glucose, those 2 pyruvates create 2 acetyl-CoAs. So each turn of the citric acid cycle creates 3 NADHs that would lead to a total of 6 NADHs. And we also have FADH that's produced. And then we have 2 direct phosphorylation steps, or this payout directly of 2 ATP. Okay. Now, from all these different coenzymes, how much ATP do we get for each step? So remember, those 6 NADHs will be converted into 15 ATPs, right? Because we take 6 multiplied by 2.5. And then the FADH, those 2 will be converted into 3 ATPs, because remember, it's 1.5 ATPs per FADH. So that means from the citric acid cycle, combining both the direct payoff that we get from the creation of ATP, as well as the indirect payoff that we get from the creation of these coenzymes, which are converted to ATP through the electron transport chain, there are 20 ATPs produced in the citric acid cycle. So that means the total yield of this overall reaction, where 1 molecule of glucose is combined with 6 oxygens to create 6 carbon dioxides and 6 waters, we have a payoff of a total of 32 ATPs in all of those steps combined: glycolysis, pyruvate catabolism, citric acid cycle, and the electron transport chain. 1 molecule of glucose would yield 32 ATPs. Okay. Yeah, so this is just, again, kind of a summary of each one of those. And again, the whole goal is to be able to figure out how much of each of these products are obtained. Actually, this slide right here does it really well. I sort of had designed that slide to go through and do this exact same thing, but this sums it up pretty well. For each of these metabolic pathways, you want to know how many coenzymes they produce, how many is a direct yield of ATP, and then factoring in the electron transport chain, what the total amount of ATP you would get from both direct and the indirect, what that would be. So you want to know that for each of these different metabolic processes. Being able to fill this chart out when it was empty is like the most important part. Again, you don't have to know the individual steps of the citric acid cycle when the NADH is produced. What's important is that you know per turn of the citric acid cycle, 1 ATP, 1 FADH, and 3 NADH. And per glucose, you will have 2 turns of the citric acid cycle. Okay, so these are a little summary, and how we got those final values of ATP is because per NADH molecule, we're going to make 2.5 ATP, and per FADH molecule, we're going to make 1.5 ATP.